Okay, so now I'm recording. The third exam is coming up during week 11. So I believe if that's going to be on a Friday, that would be November 11th for that exam. And that material will start with chapter 7, which is on light, properties of light, and looking at chlorophyll and other pigments all the way through the light reactions. It will cover the C3 cycle and the PCO cycle. I still have to make up my mind where I'm going to put the C4 and CAM. I don't know. It might be on exam 3. I'm thinking I got to finalize that, but a couple of weeks out now. So around November 11th, it looks like we'll have the third exam. I am thinking of holding a review session earlier that week, or maybe even that weekend prior, like a Sunday night or something. I don't know. Would that be useful? Okay. I've done it several times in the past, and sometimes a lot of students show up and sometimes hardly anybody. But I imagine by the time that weekend rolls around, well, hopefully you've been studying a little bit on this material coming up for exam 3, but I know this is a rough time in the semester for everybody. So I know that some things can slide, and that's why I tend to like to have a review session, almost a whole week ahead of the exam. In case it's been a while since you've looked at this, you'll have, we'll meet, we'll go over it, and then you'll have several days to get it cemented into your long-term memory. It takes time to put things into your long-term memory, so you have to be looking at this material frequently to get that to happen. And I don't mean just looking, like actually interacting with it and studying it and learning it. There was something else I was going to say. Oh, I was just going to ask if like Sunday early evening, does that seem to work for people? Do you think like 6-ish, 5 or 6, something like that? I'll take a poll as we get closer. And then what I usually do is find a room, this room or some room that we can use and meet in person. Okay. Well, we're on to a new slide set. Is that visible? Yeah, it's visible in EDUCAT. The signals and signals transduction, I'm pretty sure I posted that. So that last slide set we just covered on Wednesday was pretty short, just looking at photoreceptors. Now I want to look at kind of taking it a step further. Like once those photoreceptors absorb that light energy and they shapeshift, then their job is to send that signal into a signal cascade of some sort. And this is not a molecular biology class, so we're not going to get into the nitty-gritty. However, there's some basic things that you need to think about. And obviously for plants, being able to assess their environment is absolutely crucial, because they don't have the luxury to run away or move away from a habitat that's no longer serving them. They need to deal with whatever is getting thrown at them. And so being able to assess their environment, in fact, the number of genes that they devote to this is far higher than what we have in terms of assessing our environment, because we, again, have the luxury to alter our environment or move away very easily. And so this is a constant strategy that they have to employ. And so they do have very sophisticated mechanisms in place to do this. And like I said, we can't get into the details of this, but there's some basic ideas. So of course, what they're assessing are all the different signals that they're being exposed to. And the way they tend to do that is through the signal docking into a receptor protein. And so we think about receptor proteins. And then once that happens, that's usually, not always, but usually on the cell surface that interaction occurs. But then you got to get that information into the cell. And so we talk about the fancy word is transduction, which usually means that you're transducing means it's moving. And then often we're amplifying at the same time that it's moving. And so you'll see that there could be this signal cascade that kind of ramps up the signal So by the time you have the plant starting to shift into a response, it's a very definite response. And so when we say response, it's usually due to some shift in gene expression to allow that to happen. So when they talk about the transduction, basically it's getting that information, so that signal that docked into the receptor, now we have to kind of convert that and get it into the cell and get it so that the plant can understand it. So it is often a conversion in a way, because it's not like the signal necessarily comes straight in. And then there's usually an amplification of it, and that can occur in a variety of different ways. We'll mention a couple. The broad term for how these signals get amplified is the use of second messengers. And they used to call them secondary messengers. So if I say that, that's what I learned. And so I tend to revert sometimes and say it the old way, but I think most current readings, they shift it to just second messengers. And so we'll look at a few of those and think about how they are allowing for that signal to really be heard, so to speak, by the inner components of the cells. And usually we're thinking about the shift in gene expression. So this is a nice visual image showing you a variety of options, some that are very quick, like what you see on the left, A and B, with the wonderful Venus flytrap plant and that poor hapless fly that happened to land in that trap, and then that snapped shut. I think all of us are very familiar with that, seen plenty of video of that. You can buy Venus flytrap plants for your house. By the way, you don't have to feed them flies if you don't want to. You can. I used to do it just because when my boys were little, they thought that was pretty cool. But you don't have to do that, because usually the soil that they provide for the plant, that when you purchase it, the soil gives them everything they need. But in a natural habitat, Venus flytrap are lacking in nitrogen, and so they love the nitrogen coming from the exoskeleton of the insect. Here you see that beautiful sundew plants. If you ever see sundews in a boggy area around here, good eye. You have to have a good eye for that. And they're very petite plants, so they're there, but you have to really hunt for them. And so those are very quick responses. On the other hand, you see here in figure C, that's a very slow, over many years response to wind. So the wind in this image is coming from the left off of a cliff face. I think this was taken on a cliff top, where you have strong, consistent winds coming in that direction. And you can see how the tree has adapted to allow the wind to just kind of flow off of it and try to minimize damage to the strong wind forces. There's a tree like this on the island, Presque Isle. Mackinac Island. Why did it come up with that? Presque Isle Island, so to speak, out there. There's one of the trails that leads out to the cliff overlooking the water. Does anyone notice that one? It's there on one of the trails. You can't miss it. I have a picture of one of my boys on that, sitting on it. So there was probably some obstruction in the way over time that caused this. Although I know that some Native American people will do that on purpose. It's a way for them to mark their trail. They would actually get the trees and young saplings to grow in a very peculiar manner. And that was a visual that they were there, and it was leaving visual information of who was there. So the Sioux would do that. Oh, I know. Yeah. So I don't know if maybe that was the result of somebody doing that, or if often it's an obstruction that's in the way. Yeah. Yeah. Isn't that something? Yeah. Yeah, it's so cool. Plants are amazing that way. They're so adaptable. I was going to say for some people that there was a spot that's really tall rock. And what's interesting to figure out how you find all these trees, they all to that? The rock. Oh. Yeah. Yeah. Neat. Yeah, I've seen other trees in various other hikes I've taken in the area that are like that too. And I always think, I wonder if that was somebody who did that or if it was just an obstruction in the way. But kind of reminds me of that poem by Robert Frost, Birches. If you ever want to relax and read a good long poem, that one's how he likes to climb up to the young sapling and then have it bend over and, you know, kind of leap off of it. It's a good poem. Anyways, so this figure here, what I would like you to know the step-by-step of this. All right, so this is the big picture idea of how you have a signal, some kind of signal in the environment that will usually dock into some kind of receptor protein, and then that interaction causes the transduction of the signal into the cell. So then you get that cellular signal transduction and amplification often, all the way in until you get some kind of response. Usually we need a shift in gene expression of some sort in order to get the behavior to respond, you know, the plant to behave a certain way in response to that signal. And then on the far right, they're just giving some examples of what does that mean. You know, so the environmental signal could be touch, light or temperature. I was just reading something the other day and I just liked the way the author phrased it. And it's true. They said, plants know you're there. They even know what colors you're wearing in some respect. So when you're standing next to your houseplant knows you're there. I think that's a cool thought. And when you touch them, they have all kinds of touch receptors all over their body. They're exquisitely set up for touch. And so if you touch them, they definitely can feel that. Okay, so then here, the receptor, we'll talk about a couple of these, not all of them. Second messengers you see are on the signal amplification idea. We will see that phosphorylation often comes into play to help relay the signal further. It's usually a transfer of a phosphate group. That's a very common strategy. We can also have repressor proteins being broken down in order to release that repression. So sometimes it's a release of repression, not necessarily this induction of something. So we'll talk about that. Hormones definitely play a role. So we're going to get into all the different hormones. But we also need to talk about electrical signaling, and plants definitely have that ability. That's been proven. Although when that first came out, a lot of scientists are like, no, plants can't do that. And now we know, yes, they can. So they use electrical signaling. And then the response, you can see gene expression ideas, some kind of alteration in gene expression. So this slide, big picture ideas, please spend some time learning. So here are just some examples of parts, the different parts that we were just looking at. So with this, they're just showing a plant cell, and then you're seeing some receptors are in the cell membrane. So brassinosteroids are a type of hormone that we'll talk about. Mechanical stimulus is when you touch a plant, they have mechanosensitive receptors in their cell membrane. Here you're seeing different hormones being played out on this image of citric acid, cytokinin, ethylene, jasmonate or jasmonic acid, gibberellin. So we'll talk about all of those. You can see that some receptors are actually not in the cell membrane. They're actually in inner membranes. So in this case, the endoplasmic reticulum membrane. So it's not 100% in the cell membrane that you find those receptors. And then there's some receptors also that they found that are just kind of floating around. So and then here they're showing the nucleus, how you can tell that there's certain receptors in the nucleus for some of these hormones. So there's some good variety. Even the chloroplast has some receptors. So yes, the cell membrane has a lot of these receptors, but it's not 100%. You can find them in other areas of the plant cell. And then what they've discovered is calcium plays a much bigger role as a second messenger than they thought possible. So the original hypothesis actually was that calcium wasn't such a big deal for plants. They didn't think it served as a second messenger. And then they started doing the more sophisticated tests, experiments, and they found out just the opposite. And the more they look into it, the more it seems calcium is very heavily involved as a second messenger. Shifts in pH can actually serve as a way to be a second messenger. And so sometimes you'll see during a signal transduction scheme that you'll see a flood of protons, because remember, that's how we assess pH, right? It's the negative log of the proton concentration. And so basically what this is saying is it's the proton concentration that you follow when you're looking at that signal cascade. So I'll show you an example. And then even they're finding, I went to a conference where they gave a whole seminar just on this idea, that they're now discovering that there's membrane lipids that will kind of pinch off from the membrane and serve as a second messenger during these cascade processes. I'll show you a couple examples of those. And then reactive oxygen species actually serve as second messengers. So I want to raise the point here that very often when we talk about reactive oxygen species, it's a very negative thing. We talk about how bad that can be because they're reactive oxygen and they react with things they shouldn't be reacting with and causing problems. So they can cause cell membrane damage and DNA damage, protein damage, and all of that is true. However, in plants, they're finding that reactive oxygen species is both good and bad. It just depends on the situation you're looking at, whether it's a good thing or a bad thing. And so again, this is kind of like too much of anything can be bad for you idea, where sometimes you do need a little bit of these ROS molecules in order, in this case, to help with signal transduction. So we'll come back to that idea. Whoops. So here is a very messy slide with a lot of stuff happening. I want to point out just a few things that I want you to know from this. So here they're looking at primarily calcium ions and pH shifts going on. So the calcium are the yellow balls. So if you just pan your eyes across this image, I think you can see a lot of yellow balls in this image. And so that's one thing to catch. Calcium's a big deal. So calcium is being used heavily in a variety of messaging systems. Here, the CA, that stands for calcium, and then the capital M is, do they have it spelled out? Calmodulin. There it is, down in number 3, activated calmodulin, CAM. So calmodulin, that word I want you to know. That is a protein that interacts with calcium. So calcium will bind to calmodulin. And we're finding that that is very, very important in the signal transduction process. So calmodulin, I would like you to know the basic idea, oh, that binds with calcium and calcium is a second messenger. So they're showing here in step 3 that they've labeled that you have that calcium, it's bound to the calmodulin, and then they're trying to show how that complex then can interact and keep sending the signal. We're not going to get into details on that. Where's the other thing? Let's see. Well, they're showing protons getting pumped across the membranes, both in the cell membrane and then in the vacuole. So they don't show that too dramatically here, but that's the other thing is think about proton shifts going on, whether a proton is shifting out of the cell or into the cell or maybe into or out of one of the organelles. So just that shifting of the protons. And often that has to be done through expenditure of ATP. So if you're going to pump protons across a membrane, you typically have to use ATP if it's going across a gradient or against a gradient, I should say. Okay. As you can see, they don't know a lot about, there's still a lot they don't know. The dotted line, they're like, we don't know. So they're still working on it. Here are some chemicals that can be derived from membrane lipids. And so again, it's just kind of like pinching off some of the cell membrane. And then there's these small molecules coming from the membrane. And so these can be signals or second messengers, depending on the situation. So diacylglycerol, inositol triphosphate, phosphatidic acid. So molecules actually coming from the membranes can actually serve as either a signal or as a second messenger. We'll talk a little bit more about this idea when we get into plant defenses. So sometimes when an insect, let's say, is chewing on the tissue of the plant, some of the membrane material will break free, and that will actually, it serves as a signal to the plant. Eek, I'm getting attacked. My membranes are getting chewed into bits. But they can then act as second messengers to help the plant to start to defend itself. Here's the ROS idea, reactive oxygen species. So all of those listed on the top green bar there, oxygen, not so much, but then the superoxide anion, peroxide, and hydroxyl radical are all reactive oxygen species. So they have the ability to interact with other molecules near them, and because they have those extra electrons, they're unstable, and they start grabbing electrons from nearby molecules, and it's just this cascading domino negative impact of interacting with these molecules that creates more and more free radicals, and it just kind of snowballs on itself and can cause a lot of damage to your cells and tissues. It's happening in plants, it's happening in us. This lower image where they show oxygen accepting stepwise electrons, so one electron at a time. This is what's actually happening in your body right now in your mitochondria. So you have oxygen sitting at the very end of an electron transport chain process, and as those electrons are zinging down to oxygen, it'll grab one electron and become the well, it's a ROS. I don't know if that's the superoxide anion. And then another electron becomes hydrogen peroxide, another electron hydroxyl. So all of these intermediate stages before it becomes water, these are all free radicals. So in your cells right now, in your mitochondria, if this isn't going smoothly, sometimes what happens is these free radicals kind of go on a rampage and have a mind of their own and start causing issues. And so your body does this every second of every day. And this is one reason why if you don't die from anything else, you'll die from oxygen, essentially. It's because over time, these free radicals, these reactive oxygen species can cause aging and eventually death if you don't die from anything else. So pleasant thought. We absolutely have to have oxygen to survive. But in the end, it can get you. So kind of an interesting process here where it's, a lot of people think it just goes right away from oxygen to water, but it's actually a stepwise reaction as being shown here. Okay, so here they're looking at the idea of reactive oxygen species. And so this is trying to show you that, this previous slide, I just told you reactive oxygen species are bad. And yes, that's true. Here is showing reactive oxygen species is actually good. So you're seeing both sides of it. So this is actually showing that plants can use some of these reactive oxygen species as a signal and a way to respond to different stresses. A lot of what you're seeing here is mostly abiotic, so environmental stresses that plants have to deal with because they can't get up and walk away. So they will routinely deal with many of these on a daily basis, and certainly through their lifetime. And so you can see that all these abiotic signals are impacting the production of reactive oxygen species. And when those get made, it is a way for the plant to then kick into a stress response, an appropriate stress response that will help them. So in this case, this is good. The only downside here is you see this red line coming off to the right with that 90-degree little thing. Whenever they draw it like that, that means something stopped. All right, they're trying to say it stopped. So they're saying reactive oxygen species definitely slow down growth or could stop it. And certainly the development into the full, like it could stop flower into fruit production, for example. I also edited this picture, this figure, because we will very soon start talking about how plants protect themselves from pathogens. And so reactive oxygen species also come into play under those conditions. So here we're seeing the good side of reactive oxygen species, that it's actually necessary for the plant to adapt and shift their behavior appropriately. Okay, so hormones play a big role in the signal transduction process. And so we're going to get into hormones. I know that you've thought about them already with lab experiments that you've done. So some of this might sound familiar, but let's get into some of the big picture ideas with hormones. So again, it's a way for a multicellular organism like a plant to be able to communicate with distant parts of its body. And so certainly intercellular communication. And there's some differences if you've taken human phys or animal phys, how hormones work in animals is a little different for plants. So in plants, you can find one hormone being produced in many different parts of the plant. It's very common. You can find that a hormone in a plant can act locally, and it can also act at a distance. And at least most animal hormones that I'm aware of, they only act at a distance. And so here we find plant hormones can do local and distant impacts. There's a concentration dependent effect. A little bit of the hormone gives a little bit of a response. You get an optimum concentration of the hormone for an optimum response. And then you can actually push it into a negative idea where too much hormone can actually create problems for the plant and be a negative thing. So concentration dependent. Each group of hormones that they've discovered often have a wide variety of impacts. Personally, I would never want to be studying hormones as my career. I'd be bald by now. I'd be pulling my hair out because it's just, I give a lot of credit to people who do study this because it's agonizing in trying to tease apart what does this hormone really do? Because there's so many hormones that interact with each other and they impact the same response. So it can get extremely challenging to tease all this apart. And you have to have good technology, good funding. Okay, so here's a general scheme of thinking about how hormones are impacting the signal transduction process. So here you're seeing kind of the kickoff on the top where you have some environmental signal. And then that usually will kick in hormone synthesis at that stage. Once the signal has been received, you'll start to see that. And then you can see, okay, yep, the hormone gets synthesized, and then it often can act locally, but it often has to get transported. And that can be perhaps through the xylem, perhaps through the phloem. It can be from cell to cell to cell to cell through the plasmodesmata, or we'll talk about some of those. And then there's usually a receptor eventually that the hormone will dock into at some part of the body. And then that will kick in a signal transduction at that site. Then you see off to the right here, threshold. What they're saying is that eventually, well, here's a feedback where you can either ramp up hormone synthesis, and it's just context dependent, like you have to take it case by case, whether it will be, let's ramp it up, or you might have a feedback where it says, okay, slow it down, we don't need this signal cascade anymore, we're done. And so they have to have a way to both ramp up hormone and then ramp it down. It makes sense that there's got to be a gas pedal and brake pedal, so to speak. And then you see response termination. That's when you really make everything just, okay, go quiet, no more. And so there's a variety of ways. You could pump out the hormone, efflux it out. That's a mechanism. You could break it down. So catabolism just means break down the hormone. Or you could stuff it somewhere, put it in a vacuole or somewhere. Or we'll talk about conjugation. That's where you usually attach some other molecule to the hormone, and that's like putting a blanket on it and telling it stop. All right, so we'll look at those. So lots of finesse to both ramping up and quieting down the action of the hormone. Here is just an overview of most of the hormones that we'll talk about are on this image here. Pretty much all of them, I guess. So we'll look at all of these in turn. Some are more well studied than others because we've known about them for longer. Probably the newest ones on the scene are jasmonic acid and brasinosteroids and strigolactones. Those are probably the ones that we need to learn more about. They're relatively new finds. Okay. So we're going to start with the top left, indole 3-acetic acid, which you've learned about in lab. that it can have a variety of impacts on plants. It was actually discovered, although they didn't name it, but they discovered this one with Charles Darwin and his son, actually, his son Francis. They did their initial work on this, and that was with, they actually wrote a book, The Power of Movement in Plants. And so they were looking at plant movement, and they just asked the question, how does that happen? You know, why does the plant bend toward the light coming through our window in our dining room or living room or whatever? And so they had a home botany lab all set up, and apparently they did a lot of research in their home area. And so that was their simple observation, which led to the whole kickoff of hormone research. And so indole-3 acetic acid is the most abundant made by the plant. It seems to have the most dramatic impacts on the plant, but there's other types of auxins. So really that should have an S on it, auxins, because there's multiple versions. It's just that indole-3 acetic acid is like the one we tend to focus on when we talk about this. Okay. This diagram is trying to show that it can be conjugated or free. So here in the yellow box, it's free indole-3 acetic acid. It's all by itself. But you can see in the blue boxes that there can be molecules that could attach to it. Then we say it's conjugated. So the blue boxes is a conjugated form of indole-3 acetic acid. When any hormone is conjugated like this, it's usually some kind of sugar or some small molecule that they'll use. That's quieting the hormone. The hormone won't work. It won't have its normal impact if it's conjugated. It's like putting a blanket on it and telling it to go to sleep. If you cleave off that molecule, then it becomes active and starts doing what it's supposed to do for the plant. So this is a very simple but effective tool by the plant. Just stick a molecule on it to quiet it down. But then it's ready, it's already synthesized, ready to go on quick notice if need be. All they have to do is cleave that off and boom, there it is. It's ready to be used, rather than having to synthesize it from scratch, which would delay the response. So it's a very effective way to have a very quick turnaround in having a quick response, getting that indole acetic acid free. So this is work that Darwin and his son did, where we're looking at phototropism, where it's that unidirectional light that they're bending toward. And they just did some really simple experiments in the late 1800s where they noted that that was happening, and then they decapitated the top of the corn seedling and did the same experiment, and they didn't see any bending. So they concluded that there's some kind of something going on in the tip that's allowing the plant to respond and bend. Then they said, well, what happens? Maybe us, decapitating it did something. So they said, well, let's just cover it with a light-proof cap. And again, no bending. So they knew light needed to be involved into this bending response. And then you can see, I won't go into more detail, but lots of other scientists came afterward and did more and more research. And again, the idea is that when they got it all pieced together after many decades of work, what they realized is happening is that auxin is being produced in the apical meristem region of the shoot. And when you shine the light, in this case, the light is coming from the right, what's causing auxin, the light is causing auxin to shift to the dark side. All right, and so then it starts flowing. All the blue is auxin. You can see it's flowing down the dark side. A very nice close-up here on the right, where it's just the dark side, getting that flood of auxin. And so what's happening here on the dark side, auxin is saying, grow, grow. You know, I want cell division, I want cell enlargement, grow, 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 grow. But the lit side is not getting that signal. So it's, again, this asymmetrical growth that is being caused here by this asymmetrical flow of auxin. And so auxin is flowing down the dark side. I can't help but think about Darth Vader. My two sons loved the Star Wars series, and I've lost count how many times I've watched those movies with them. So anytime I talk about auxin and going to the dark side, that jumps into my head. So if that helps you remember what's going on, use it. certainly helps me remember. So it's, again, this asymmetrical flow of auxin, and auxin is known to stimulate growth. And so it's this asymmetrical stimulation of growth. And if you keep doing that, you can imagine how, yeah, that would cause the seedling to bend like that toward the light. And of course, that's good, because what's happening is usually then the leaves are being presented to that flow of light at a 90-degree angle, which is what you want. So Charles Darwin and his son Francis started that all off, and they didn't name it, but later people came to name it auxins, and they figured out indole 3-acetic acid and all the other ones. And so they started hunting, like, where else is auxin? Where are they made? By the plant. The shoot apical meristem, the root apical meristem are good sites for that. Anywhere there's rapidly dividing tissues, you'll find hormone synthesis and action. One thing about auxins, though, that's peculiar compared to, I think, every other hormone they've discovered, it exhibits polar transport. So what that means is auxins flow from, like, the shoot apex down the stem. Doesn't flow the other way. I'll show you a picture here to try to demonstrate this. And so even if you turn a plant upside down, it will still flow from the shoot apical meristem down the stem, even though you've turned it upside down. I'll show you a picture to get across. So this is very odd. It also tends to move from cell to cell in a pretty kind of oddly complicated manner. You think, why don't you just use the plasmodesmata? That's the fast way to go from cell to cell, and auxin doesn't do that. That's another really kind of odd thing, and it requires energy. ATP is needed. So I'll show you some images to get at this in a minute. So, and it needs special carrier proteins to get the auxin through the cell membrane into the cell. It's just complicated. It's like, why would you be so complicated? Just go through the transmembrane the plasmodesmata would be the easiest. So there's some interesting ideas about why it does it like this. It's independent of gravity, so what I mean is you can turn the plant upside down and it still will have auxin flowing from the apex of the stem down the stem, even though it's upside down now. Long distance transport, if it wants to go long through the body, it usually goes through the phloem. And that makes sense. That's a nice highway system, internal highway system. Use the xylem or phloem to get to distant parts. And that doesn't seem to involve any ATP. It's just flow. It's important to also realize that microbes that coexist long-term with plants, many of them also can produce auxins. And many of these bacteria are good, like they're helping the plant by doing that. Unfortunately, some pathogens also can do this and create problems where they overstimulate the plant and cause problems. But many good, beneficial microbes can make auxins. It's identical to what the plant makes. They make it too, and they excrete it. And so the plant gets bathed in a little bit of extra auxin produced by their partners. And that can be very good. This is the independent of gravity idea. So the cutting on the left, they put it upside down and then they just watch to see what would happen. The cutting on the right is the normal orientation. And what they're trying to show is that, you would think you put it upside down, that the top part would be the leaves, the bottom part would be the roots, and it's not. And so if you take a plant and put it upside down, you still have whole from the so-called apex, the shoot apex, to the basal end, which is the base of the stem. It's kind of weird. Oh, I didn't have it in here. Where is it? Oh, here it is. This is the other idea, polar transport of auxin. This is further down in the slides. So here it's this complicated thing of we have three neighboring cells that are being shown here. And so you're seeing the indole acetic acid coming out of the top cell, and it is getting into the second cell here through a transport protein, and then it flows until it docks into these receptors and gets transported out and then gets into the next cell. So it's just an ATP is needed to get this to happen. So this is actually made some scientists think a little bit more about, could auxin be serving as kind of a neurotransmitter type function? This is very controversial, but there has been some intriguing studies done where they are showing the analogies between how auxin is moving from cell to cell like this. And it's similar to how neurotransmitters work from one nerve cell to the next. So there's been some very interesting ideas. You may have heard perhaps that some scientists are looking very closely at roots, and especially at root tips as kind of analogous to brain function in terms of how neurotransmitters and neurons work. They're starting to look at that in the root tip region. I know that sounds really weird, but if anyone wants to talk more about that at some point, I can direct you to some papers or chat with you. Very controversial, but very intriguing ideas to think about. Okay, all right, where's the other one? Okay, well, let's go into this one. Let's talk about cytokinins. I'll be introducing especially the classic 5 hormones, and then I'll talk a little bit more about what some of them are doing. Here, the cytokinins are another group, kinetin and zeatin. So zeatin is the one that actually the plant makes, although we talk a lot about kinetin, but kinetin is synthetic, actually. We make it, humans make it, and it's related to zeatin, has the same impact. It's just more stable to work with. So we tend to focus a lot on kinetin because zeatin is a pain to work with. But plants make zeatin version of cytokinins. Bacteria can make it too, the good ones and the bad ones. Insects as well. And so some insects, like wasps, there's certain wasps, I can't remember the species, I'm not good with insects, but they will actually sting the branches of a tree over and over and over. And what they're doing is they're injecting zeatin into the stems, the branch cells, and it stimulates those plant cells to make like a gall or a tumor. And I guess the wasp kind of like lays its eggs in there or has a home in there. Kind of freaky. And so that's kind of one of the more extreme examples of a non-plant organism making a plant hormone. And so it's doing it, the insect's doing it solely to benefit itself. So the plant doesn't die from that, but it's, it's all for the insect. Bacteria and fungi are also known, both the good and the bad. And also there's a particular group of, there's a certain species of bacteria that also does stimulate the plant to make a gall. And the Agrobacterium is known to do that. That's a bacteria. So it's kind of interesting that insects have kind of co-opted that ability too. Benzoyluminopurine, I think, is what we worked with in lab when we made up cytokinin solutions for you to soak your, remember you did those leaf discs where you took spinach leaf discs and put them into different solutions and then put them in the cupboard and let them soak for a while. And then you were looking at what did that do to the ability of the tissue to stay fresh or to go through senescence. Again, free cytokinin is active. If it's bound up or conjugated, it's quiet. And again, it's usually a sugar molecule that they'll attach to the cytokinin to get it to stop having its impact. So in lab, we were looking at how the benzoyluminopurine type of cytokinin was retarding senescence. And so there's a threshold level where you can see a certain concentration will really preserve and keep the tissue fresh. What you see on the left and the one on the right definitely was that's severe senescence that you're seeing here. And so we were looking at that in lab. So cytokinins are both auxins and cytokinins, by the way, both of those hormones. They have yet to make a mutant plant that can't produce. It won't work. They cannot create a mutant that can't produce auxin or cytokinin. The plant dies very quickly. And so those two hormones, auxins and cytokinins, are absolutely critical for the plant. And we use auxins and cytokinins all the time in plant tissue culture, which you'll do next week. Hopefully everything's going to go fine. And so we actually mix in different concentrations of those two hormones in order to stimulate different types of growth in plant tissue culture. And so here you're seeing on the bottom area, that's what's called a callus. You'll learn about that in lab. It's just this clump of undifferentiated tissue. It just looks like a tumor almost. We call it a callus in the plant world. And then if you give these callus, if you expose them to certain ratios of auxins and cytokinins, you can get shoot and leaf development, you can get root development, and you can stagger this where you trigger, just get the callus going. And then you could say, okay, now start forming shoots. Okay, now start forming roots. Or you can do it all in one fell swoop. It just depends on the protocol and what you're trying to do. So we'll play around with that in lab. You'll start to play around with these hormones and look at what happens when you expose the plants to this. So you'll be cloning plants in lab, hopefully next week. All right, we're going to stop there and continue on next week with the other hormones and saying a little bit more about how they impact the plant.